Hey everyone, welcome back to MedMe. Today we're going to be talking about chromatin. So before we get started, I think it's important to kind of review the structure and function of chromatin. So if we take a look at this picture on the slide here to the left, we see a couple black circles with a couple blue and purple squiggly lines through them. So let's try to imagine these black circles without any squiggly lines. Those are depicting histones. So histones are comprised of proteins that are often called core proteins. So these core proteins that make up histones are H2A, H2B, H3, and H4. Now, something that they can trick you on a test is that they'll say, which one of these is not a core protein that makes up a histone? And they'll list H2A, H2B, H3, and H4. And then they'll also add H1. They may even say H1 linker. So if you get this question on the test, H1 linker is not a core protein. So make sure you keep an eye out for that and you really know the structure of the core proteins of the histone. Now, taking a more deeper dive, how tightly or loosely DNA is wrapped around these histones depends on whether they have undergone acetylation or deacetylation. So just to review, we know that DNA is negatively charged, and this is due to its phosphate backbone. Whereas histones are positively charged, and this is because histones carry a lot of lysines, which lysines are an amino acid which carry a positive charge. So the DNA is attracted to the lysines on the histones that make up the histones. So let's take a closer look at the process of acetylation and deacetylation. And I want to clarify, when I'm talking about acetylation and deacetylation, I'm referring to processes that happen to histones. When I'm talking about methylation, this is a process that is happening to DNA. And I know that sounds kind of redundant, but it's really important that you know that acetylation and deacetylation refer to a process for histones, methylation is for DNA. Okay, so moving on. When histones are acetylated, this means that acetyl groups are added to lysine residues. This neutralizes the positive charge that histones carry. It decreases DNA binding. The chromatin actually opens, and this can increase transcription and therefore increase gene expression. Now, for the process of deacetylation, this refers to removing acetyl groups, which restores the positive charge that histones carry. It increases histone binding to DNA, and it decreases transcriptional activity and therefore decreases gene expression. So that's a very complicated kind of topic to understand. There's a lot of moving parts there, but I will go into this process in depth in another video. So if you don't quite understand it yet, don't worry, but make sure you're kind of reviewing it before then. Now, when histones are acetylated, as we just talked about, this increases transcription. And this is referring to euchromatin. This is transcribable DNA. And if we look at this kind of histology picture I tried to make myself, it appears lighter on electron microscopy. And an easy way that, or like a trick that I use to help remember like euchromatin versus heterochromatin is that euchromatin is usable, meaning it's transcribable, it can be involved in gene expression. Whereas heterochromatin, it's very condensed DNA. It's deacetylated, it's DNA that's tightly bound around the histones. It's not transcriptionally active, therefore there's decreased gene expression. Now, heterochromatin usually appears dark on electron microscopy, and it's usually found at the periphery, which we're kind of seeing in this picture here on the histology kind of slide that I tried to depict. So that is basically an overview of heterochromatin versus euchromatin, and it's important to know the processes of acetylation and deacetylation when you're referring to heterochromatin and euchromatin. Like I said, it's okay if you don't understand this right now. This is something that sometimes takes some time to wrap your head around, but I will go into much more detail on this process in further videos. So please stay tuned and make sure you like this video and subscribe and get the slides that will be found in the description. Thank you.